Hi, this is Dr. Muscatello, and we are going to dive into Chapter 4. This is one of the chapters that has really sort of exploded in immunology, and there's a lot of updates and new and exciting stuff in this chapter. So why don't we go ahead and get started? The innate immune response is responsible for really our first line of defense against pathogens. It helps to actually keep us alive while the adaptive immune response gets up and running. All of the innate immune defenses are present at birth. They don't really change with age. They never really get any better as the adaptive immune system does. And they have an extremely limited diversity for antigen. If we're making a comparison, the innate immune system can recognize maybe less than about 1,000 different types of molecules versus the adaptive immune system, which we know can recognize millions of different types of molecules. Based on the different types of pathogens that we can be exposed to, the innate immune system essentially has two major ways that they can deal with these pathogens. We have the inflammatory response that helps us to eliminate microbes, and Dr. Ali is going to talk to you about that in just a little bit. And for our intracellular friends, we have molecules like interferons and cells such as natural killer cells that help us to eliminate viruses and other sorts of intracellular infections. Okay, let's talk about how the immune system deals with pathogens once they gain access into the host. There are lots of different portals of entry that we're going to talk about in just a little bit that pathogens have to breach. If those barriers are breached, the organisms can gain access into the tissues. What awaits them in the tissues is an entire army of your innate immune system consisting of phagocytes that can certainly help try to eliminate the pathogen. There's an inflammatory response, lots of cytokines we're going to talk about, and Dr. Ali is going to talk to you about that in just a little bit. Oftentimes, the innate immune system is fast enough and good enough to eliminate the pathogen, but of course that's not always the case. And sometimes the tissues are breached and the pathogens gain access into the blood. Now, if we're talking about adaptive immune system, of course, we have antibodies and other things in the blood. But as far as the innate immune response, we can have alternate complement pathway activation that can help with opsonization and removing immune complexes and things like that from the blood. But if those all fail, that's when the adaptive immune system will take over. And we're going to definitely talk about that in some upcoming chapters. There are several components to the innate immune response that are going to be really important for dealing with pathogens. We have physical and anatomic barriers, physiologic barriers that Dr. Ali talked to you about early on in Chapter 1, the innate cellular response, which deals a lot with phagocytes and phagocytosis, and of course inflammation. I'm going to introduce you to the physical and anatomic barriers and some of the physiologic barriers, and then Dr. Ali is going to take over and talk to you a little bit about the innate cellular response, which really plays nicely into the acute inflammatory response. Let's first take a look at the anatomic barriers. There are obviously several different portals of entry for pathogens, but the largest are the skin, the respiratory tract, and the GI tract. All of these surfaces are lined with cells that can sort of help as far as resisting microbes. There are molecules such as defensins and interferons that we will talk about in a little bit. There are also some unique T cells that we call intraepithelial lymphocytes or gamma delta T cells. These are sort of a smaller subset of T cells. They're actually considered part of the innate immune system because they don't have quite the extensive diversity that our alpha beta T cells do. They also can develop extrathymically, so they're really sort of completely different. And mostly, if we're talking about T cells, for step 1, you're going to really want to think about the alpha beta T cells. Those are the ones that they're really going to focus on. As far as anatomic barriers go, the skin is an amazing first barrier. Most pathogens cannot invade intact skin, so they all have to sort of get in through a break in the skin. There are a few when we start getting into some of those nasty parasites that can just penetrate intact skin, but for the most part, skin is really a good barrier to infection. The respiratory tract is also a good barrier. There's cilia in there that help us to remove particles and microbes, even down to actually the little vibrissae, the little nose hairs that sometimes in people get a little out of control, I'm not going to lie, are there to help you sort of remove particles so you can eliminate those before they actually get into the respiratory tract. The GI tract is sort of a nightmare for pathogens. Lots of mucus in there that make it tough to navigate. Similar properties to the respiratory tract, and what you want to think about as we get into dealing with the immune system is that the mucosa really sort of is one sort of large system and really has its own sort of unique way of dealing with pathogens. And once you get a really good immune response in the mucosa, whether it be the GI tract, the respiratory tract, or the genitourinary tract, the entire mucosa is protected, and that's a really important concept for USMLE. Physiologic barriers are just as important, so let me take you through some of these. Temperature is a really good physiologic barrier. We always sort of think that when somebody gets a fever that that's a horrible thing, but of course most pathogens don't grow much beyond normal body temperature, so when the temperature gets elevated, those pathogens don't do as well. pH is also important. The skin has a slightly acidic pH, and of course your gastric acidity is such that most pathogens can't survive there. A lot of GI pathogens really just sort of cross their fingers and hope to make it to the other side. You do have pathogens like Shigella and Campylobacter that are a little bit more acid resistant and don't require as high of a dose of organisms to initiate an infection versus some infections like Salmonella and cholera where you need sort of an overwhelming dose because they're extremely acid sensitive. Different chemicals are also important physiologic barriers if we're talking about things like lysozyme, for example. Lysozyme is present in your secretions, your tears. It can break down peptidoglycan in the cell wall of bacteria. And then, of course, our interferons. We're going to talk quite a bit about interferons when we get to both the antiviral response, and then we're going to talk about interferon gamma, which, of course, plays a major role in macrophage activation.
Well, let's go ahead and pick up where Dr. Moscatello just left off with us. I want you to first put into context where all of these events are going on. And it's a very simple answer. This is occurring at the site of infection. So when we talk about the innate immune response, really what we're talking about is that primary response that we need in order to control the spread of pathogen. And in several instances, the innate immune response is actually enough to contain as well as eliminate the infection, never needing the adaptive immune response to be engaged. So let's talk about the cellular response. And again, remember, this is at the site of infection. So the cells that are going to be really important here are our phagocytic cells. Recall from chapter 2 when we were discussing our monocytes versus our macrophage. Our monocytes are those juvenile forms of our macrophage that are going to be within the bloodstream. And once our monocytes exit the bloodstream and get into the tissue, they're going to take up residence in the tissues in the form of macrophage. And our macrophage are going to hang out there essentially and clean up the mess and wait to see pathogens so they can become activated. Also within that area, you're going to find dendritic cells. So we have two primary sets of phagocytes that are always within a tissue. Now, let's not ignore our neutrophils. Neutrophils are extremely important when it comes to an innate immune response, but you need to recall that neutrophils generally are not found in tissues unless there is an infection or an inflammatory response. Rather, our neutrophils are going to be the first leukocyte that is called into the site of infection when you have activated macrophage and dendritic cells. So this is really part, not whole, but only part of the innate immune response. And Dr. Moscatello is going to talk to you about the second aspect of this, which is going to be the activation of complement. So we really need to ask ourselves, how is it that these phagocytes are able to recognize the pathogen? And go back to the basics. These cells are not specific. And I'm going to give you an example of this. So we might have macrophage that recognize, let's say, gram-negative bacteria. The macrophage are not going to be able to distinguish between, let's say, E. coli and salmonella. Rather, the macrophage is going to recognize a shared pattern between those, such as lipopolysaccharide. And how they are able to recognize those patterns, which we're going to refer to on the next slide as pathogen-associated molecular patterns, is based on the fact that they have receptors on their surface that are going to interact with the shared sequence. Collectively, we refer to these receptors as pattern recognition receptors. And again, remind yourself always that where are these receptors found? They are found on the phagocytes. And again, the phagocytes of import right now are going to be our macrophage, our dendritic cells, and our neutrophils. The pattern recognition receptors, also known as PRRs, are going to recognize different patterns. That's what the name tells you that they do. So what are these patterns? Well, we have two categories of patterns. I've already alluded to one, the pathogen-associated molecular patterns. Obviously, those are going to be dealing with pathogens, but we also have what are known as damage-associated molecular patterns, otherwise called DAMPs, which are going to be found on host cells that are either dying, damaged, or undergoing some level of distress. So this is sort of a twofold approach to phagocytosis. Now, please keep in mind that these are encoded for in the germline. They are not generated through that process of gene rearrangement or somatic recombination that Dr. Moscatello went through with you. So again, these are already in the cell encoded for. So that's why, again, we can refer to them as being part of the innate response. Also, remember the limited diversity that we're seeing here. Only about 1,000 different patterns can be recognized by these cells versus that astronomical number that can be generated through the gene rearrangement. Well, just when you thought it was safe to learn immunology again, they've gone and expanded the knowledge base. Those pattern recognition receptors are now divided into several families. The first family that we are going to talk about are going to be our toll-like receptor. Now keep reminding yourself that these are simply pattern recognition receptors, which means that they are either going to recognize those PAMPs, which are associated with pathogens, or they're going to recognize DAMPs, which are associated with distressed, damaged, or dying cells. Our first family are our toll-like receptors. And I know you're probably looking at this chart and the next one saying, really, what do I need to know about this? And I'll be honest with you, 3-4 years ago, I would have been like, yeah, don't worry too much about this. But truthfully, this is an area of immunology that is starting to get tested on a lot. There's a lot of clinical correlates now between these pattern recognition receptors and various disease states. So let's take a deep breath and sort of walk through these. You do need to know at least which of your toll-like receptors are found on the surface of your phagocytes. It's fairly easy, so just start counting up. I've got toll-like receptor 1, 2, you got to skip 3, and then 4, 5. And 6. And it should make sense that these surface receptors are going to recognize PAMPs on the surface of extracellular microbes. such as peptidoglycan or lipopolysaccharides that are found on the surface of most of our gram-positive and gram-negative. Now, please note, the LPS is gram-negative. That's a high-yield point, okay? And of these toll-like receptors that are found on the extracellular surface, the one that I'm going to tell you to commit to memory and love and embrace, toll-like receptor 4, okay? Because it does recognize LPS. The others are going to be within the cytoplasmic region of the cell. And these are going to be, let's just pick up, throw in toll-like receptor 3 there, as well as toll-like receptors 7, 8, and 9. Now notice what the intracellular receptors recognize. Double-stranded RNA, single-stranded RNA. We associate that with a viral infection. So the fact that these receptors are inside the cell also makes sense, as well as things like unmethylated CpG oligonucleotides. And those are going to be associated with bacteria that have either been intracellular infections or from bacterial infections that have not been cleared. Now, what is the activity of these different receptors? In short, these receptors act to upregulate the transcription factor NF-kappa B. And this particular transcription factor is extremely important in upregulating the expression of cytokines needed in an inflammatory response, as well as adhesion molecules and different antimicrobials. The other two families of our pattern recognition receptors are going to fall under two headings. We have our NOD-like receptors and our RIG-like receptors. Our NOD-like receptors are all intracellular. We have NOD1 and NOD2. And then we have a NOD-like receptor P3, shorthand NLRP3. And I'm going to actually give you an example in just a moment of how the NLRP3 works with other members of the toll-like receptor family. Very much like we saw with the toll-like receptors, NOD1 and NOD2 are going to increase the level of NF-kappa B, and that is going to, again, lead to an inflammatory response. Whereas the NLRP3 is going to lead to the production of what is known as an inflammasome. And the inflammasome is going to be responsible for helping to generate one of the most important primary inflammatory mediators, the IL-1 beta. Now, RIG-like receptors, anytime something's at the bottom of a chart and it doesn't get a lot of shout out, we tend to ignore it. But I do want you to give it a little bit of credit and say, hey, we have a RIG-like receptor called RIG1, and we also have MDA5. Thanks, guys, for not keeping it in the same alphabet. Okay, got to make things a little more difficult. Now, this is a cytoplasmic, just like our NOD-like receptors are, but the thing is that only thing it's targeted here is viral RNA, and its effect is really not going to be to activate NF-kappa B or to help in the production of inflammasome, but rather to cause the upregulation of interferon. To give you an example about how important these different pattern recognition receptors are, I'd like to introduce you to the inflammasome, which is part of the innate immune response. The inflammasome is expressed within different myeloid cells, and if you go back to chapter 2, remind yourself what my myeloid cells are. Hey, what about those macrophage and dendritic cells and neutrophils? And this is going to be activated when we have the presence of certain pathogens or environmental stressors. And the inflammasome is part of how we generate the inflammatory cytokines, our IL-1 beta, which we know is one of our endogenous pyrogens, and also IL-18. If you have never heard of IL-18 before, you are not alone. IL-18 is sort of showing how important it is recently in immunology. It is a member of the IL-1 family, and it tends to work in concert with IL-12, and IL-12 is going to be one of the primary cytokines that will drive a cell-mediated immune response. So you can see that IL-18 is very important even if you've never heard of it before. Looking at this image, we actually have two different pathways that have been activated. So I first want to point out to you that we have activated a toll-like receptor, and recall from the table with our toll-like receptors that our toll-like receptors are going to help activate the transcription factor NF-kappa B. And one of the things NF-kappa B is going to upregulate is the expression of a pro-IL-1 beta gene. Now, remember, if it's a pro form, that it is not activated. So we need to be able to cleave the pro-IL-1 beta into its active form. So that's going to take the other part of this process, which is actually where we form our inflammasome. So if we have the presence of a pathogen or, like I said, an external stressor, that is going to act on one of the NOD-like receptors, that NLRP3. Upon being activated, it is going to interact with an adaptor protein in an inactive form of caspase-1. They are going to organize and enrage themselves in what is known as the inflammasome. And this is going to, in turn, activate the caspase-1. And caspase-1, simply a protease. And it is the caspase-1 that will directly cleave that pro-IL-1 beta into its final activated form of IL-1 beta. Okay, so now is the time where we are going to all stop and step back and ask ourselves, how is it that I'm going to take all of this new information about these various receptors and apply it to what we are going to see on the step 1? So we have put together the table that you see, and it is going to show you different mutations found in these different signaling pathways. And what we're going to do is talk about some mutations which are gain of function, and some mutations which are loss of functions and the resulting clinical presentations. So the first mutation set we're going to talk about are those that are found in the toll-like receptors. If you have a mutation in, let's say, the extracellular toll-like receptors, which are going to be 1, 2, 4, 5, and 6, the individual is going to be much more prone to recurrent bacterial infections, and the most common clinical presentation is going to be that of pneumonia. And this should make sense. Again, they are the extracellular receptors. Now, if I am more prone to infection, that sounds like a loss of function mutation. So these mutations, again, keep in mind, that's a great way they could test you on this. What type of mutation is it? It's a loss of function mutation. Examples of gain of function mutations, we have a couple examples here that are very well documented. If I have a gain in function of the inflammasome, it means the inflammasome is overactivated, which means that caspase-1 is going to be free to produce lots and lots of IL-1 beta from the pro-IL-1 beta. And remember that IL-1 beta is a huge inflammatory mediator. So when I have a gain of function here, that's associated with several clinical outcomes, such as gout, atherosclerosis, as well as type 2 diabetes. One other gain of function mutation we would like you to be aware of is the NOD2 mutation. And with NOD2, again, just do a little review in your head. NOD2 is principally an intracellular receptor that is dealing with bacteria that have been phagocytosed, and it is going to cause an increase in the expression of NF-kappa B, which is that transcription factor that turns on other genes involved in inflammatory responses. NOD2 mutations are typically seen in forms of inflammatory bowel disease. Now, the last two receptors shown here, we did not put under the general category of our pattern recognition receptors. Rather, these are cytokine receptors, but it's good to mention these here. If you have a defect in either IL-12 or interferon gamma, you are going to see an increase in the infection rate with intracellular bacteria, especially things like Mycobacterium.
Hi, Dr. Muscatello again. This table is here to sort of remind us of some of the cells that we introduced in Chapter 2, but we are going to focus on them a little bit more in terms of the innate immune system and as a prequel sort of to the inflammatory response. Our monocytes, of course, recall our baby macrophages. We're going to find those circulating in the blood. When they differentiate into the tissues, they differentiate as tissue macrophages. And then we have our neutrophils. Dr. Ali already sort of talked to you about the neutrophils and how important they are in that early innate response, and she's going to reiterate that in just a little bit when she talks about acute inflammation. So first, let us just talk about neutrophils. Neutrophils are really sort of cool cells. They're circulating phagocytes. And we already know one of the questions they love to ask us about neutrophils is about which cells are the first to arrive at a site of acute inflammation, and that's going to be the neutrophils, so that's always an important question they like to ask. These cells, though, are sort of short-lived. These guys are the soldiers that run in, they sort of clear up the infection, and they typically die in the process. So they deliver a rapid response, but not a prolonged defense, as we're going to see with some of the other cell types, such as macrophages. Neutrophils are so cool as far as what they can do to really sort of battle these pathogens, even when they're dying, and we'll talk about this in a little bit. They extend out what are called nets, which are neutrophil extracellular traps, and they can sort of rein in some of the pathogens, and they get really sticky and makes it really difficult for the pathogens to survive. So even in death, those neutrophils are still sort of working to help us eliminate the pathogens. As we just mentioned, monocytes and macrophages are going to provide more of a prolonged defense. These cells, remember, begin as monocytes, which are released from the bone marrow. They circulate in the blood, and they become macrophages within the tissues. But both monocytes and macrophages do have phagocyte activity. As compared to the neutrophils that we just talked about, the macrophages give us a more prolonged defense when we're talking about, in particular, the acute inflammatory response. They are super important in this response because they produce the cytokines that help to initiate that inflammatory response and also regulate the inflammatory response. In addition to their role in inflammation, they are also really good at phagocytosis. They are very reliant on complement and antibodies to help them with that through a process called opsonization, which Dr. Ali is going to talk to you about in just a little bit. One of the additional functions that macrophages can help us out with is to clear some dead tissue, and in the process help us to initiate tissue repair. What's interesting about macrophages, and what's sort of been elucidated a little bit more recently, is they can actually develop along one of two different pathways depending on their function, much like we sort of talk about with T cell subsets. The two pathways for macrophage activation include the classical M1 macrophages. These are the ones that, of course, USMLE is going to be a little bit more focused on. These are the ones that we were talking about that can induce an innate immune response by the production of cytokines that help to not only initiate that response but regulate it. And they are also professional phagocytes. We are also going to talk about macrophages as far as helping us with an adaptive immune response, as they also are one of our antigen presenting cells. The alternate macrophage pathway for activation are the M2 macrophages. They are induced by different cytokines such as IL-4 and IL-13, and their main role is going to be to help in tissue repair and controlling the inflammatory response. Again, this is sort of just another review of the cells that we already talked about in Chapter 2. We're going to, again, just sort of dive in a little bit deeper into their function as far as their roles in the innate immune response. And we're going to finish up this chapter actually by focusing on the natural killer cells. And this is sort of another area of immunology that's really sort of exploded, and we've gained a lot of understanding in the last few years of how these natural killer cells actually function. Another cell that's going to be really important in not only the innate immune response, but the initiation of the adaptive immune response are dendritic cells. Dendritic cells are found in all of the tissues. We talk about them quite a bit when we deal with antigen processing and presentation to T helper cells. So they have two major functions. Part of their function is to initiate this inflammatory response that we've been talking quite a bit about. And their other major function is to initiate or stimulate the adaptive immune response. So we've talked about macrophages. I know Dr. Ali mentioned that B cells were also antigen-presenting cells, and now dendritic cells. These are going to be the major antigen-presenting cells that we're going to talk about in an upcoming chapter. Mast cells are also going to be important. We're going to focus mostly on mast cells when we get into our chapter on hypersensitivity. But mast cells are found throughout the skin and the mucosa. They have a couple of different pathways for activation, including your toll-like receptors that Dr. Ali just talked to you about, and our antibody-dependent pathway, which of course is the bulk of what we're going to talk about for the type 1 hypersensitivity reaction. Natural killer cells are another really important cell of the innate immune system, and we're really going to sort of end this chapter on the natural killer cells so we can go out with a bang. Natural killer cells are found in the blood and the periphery. They are sort of the hitmen in this grand scheme of the immune system, where they essentially just directly lyse and kill cells that are either transformed or that are virally infected, and we'll talk about mechanisms here shortly. One of the important cytokines that natural killer cells secrete is interferon gamma. And then the next slide, you're going to see how important that is and how these innate cells work together to really focus on elimination of pathogens. We've now talked about macrophages, we've talked about dendritic cells, and we've talked about natural killer cells. But this slide sort of demonstrates how they all work together. We already mentioned that macrophages and dendritic cells can produce an important cytokine called interleukin-12. And interleukin-12 is a pretty potent cytokine. We're going to see it when we talk about T cell subsets. But in addition to helping T cells develop along one of those particular pathways, they can also activate natural killer cells. When natural killer cells are activated, as we just mentioned, they can produce a cytokine called interferon gamma. Now, interferon gamma is going to be a cytokine we're going to talk a lot about, but interferon gamma is sort of the primary cytokine that helps macrophages become activated. You may recall, if you saw some of the earlier videos, that we sort of talk about interferon gamma changing the macrophage into the Incredible Hulk. It goes from sort of just a nice cell that doesn't necessarily bother anyone to really sort of a killing machine. So that interferon gamma is going to be really important in this response, and we're going to be talking actually quite a bit about it in some of the upcoming chapters. So that's a cytokine that we really need to know. That's a cytokine that is also super high yield.